Hello everyone. Thank you for inviting me to the JAIMA session of Pittcon. I'm Akiko Ishii from National Institute of Health Sciences, Japan, also appointed in University of Tokyo. First of all, I'd like to appreciate the organizers of this symposium, especially for Dr. Nomura and Professor Tsumoto. The title of my presentation is same as the session title, Recent Achievements and Current Interests in Research on the Characterization and Quality Control of Biopharmaceuticals. This is the contents of my talk. At first, I would explain the current status of development of biopharmaceuticals and regulatory science research in Japan. Secondly, I will introduce the activities and research topics of Biopharmaceutical Consortium in Japan. This is a collaboration between industry and NIHS. Then I will share the outline of the new project on next-generation therapeutic antibodies. This is a collaboration between academia and NIHS. Lastly, I will mention the perspective on analytical procedure development used for quality control of biopharmaceuticals in the era of analytical quality by design. This slide shows the number of approved recombinant therapeutic protein products in Japan. Currently, we have more than 160 products. Among them, almost half is therapeutic antibodies, including antibody-drug conjugate. Their indications are mostly cancer and immune disease. The importance of biopharmaceuticals in this area is getting bigger day by day. I think this trend is similar to overseas. Here I show the examples of recently approved biopharmaceuticals originated in Japan. Tocilizumab, mogamulizumab, nivolumab, emicizumab, losmab, trastuzumab deruxtecan, satralizumab, pabinafusp. They are unique and innovative, and importantly, very useful in clinical situations. Unfortunately, products originated in Japan are limited. However, there are some, as shown here, and potential for development of innovative products is increasing, I believe. Japanese government is encouraging the development of biopharmaceuticals, including biosimilars. As one of the initiatives to promote the drug development, governmental funding agency AMED, Japan Agency for Medical Research and Development, was established in 2015. The research activities in my presentation are under the support of AMED. During the drug development, ensuring the safety, efficacy, and quality is critical. Because biopharmaceuticals have characteristics such as production in using cell culture, complex structure of the product, and less stable than small molecules. So, appropriate CMC strategy and close collaboration with molecular design from the early stage is a key for the efficient development of biopharmaceuticals. For these reasons, our research mainly focuses on the analytical method used in CMC area. This slide shows the image of risk management for safety and effective treatment. All drugs have quality and clinical issues, so mitigating the quality and patient-related risks are important. From the quality perspective, product characterization and investigating the impact of quality attributes on safety and efficacy, and selection of critical quality attributes and setting the criteria are thought as risk assessment. Then, quality control, including test of each lot, can be a part of risk control. For these steps, using state-of-the-art and appropriate analytical methods are desired. Additionally, let me introduce the word of regulatory science. Regulatory science can be said as science for scientific regulation through the research related to risk management. It contributes to ensure the quality, safety, and efficacy of the products. Regulatory science research is the mission of our institute. From here, I will introduce the activities and research topics of Biopharmaceutical Consortium in Japan. The first term was from 2017 to 2021. The second term is from 2022 to 2024. As you know, development of next-generation products and new analytical technology are accelerated. In such a case, the usefulness and/or limits of new technologies are uncertain. It is inefficient for each company to individually examine to solve such issues. Evaluation of analytical performance of the new methodology by multi-laboratory studies in the consortium is a good solution. The purpose of the study in the consortium is the establishment and standardization of new methodology used for the evaluation of quality of biopharmaceuticals. Therefore, the research topic should be not product-specific, but applicable for all or many products as fundamental methodology. This is a member of the consortium. 26 companies are involved in the group. Most of them are pharmaceutical companies, but companies for analytical equipment development such as Hitachi, Horiba, Shimazu, and Toso are the members. The research topics of the consortium are shown here. In the first term, protein aggregate analysis, N-glycan analysis, host cell protein, bioassay were the topics. As per the protein aggregate analysis, we examined light obscuration, flow imaging, dynamic light scattering, analytical ultracentrifuge in the collaborative study. We mostly focused on flow imaging methods that were thought to be useful for detecting protein aggregates. For N-glycan analysis, detailed analytical procedures were established for standardization. HCP test using ELISA-based method was established. For bioassay, fundamental requirements for relative potency evaluation was established. Now we are at the initial point of second term. The topics are again protein aggregates and O-glycan analysis, intact molecule analysis, host cell proteins, and analytical quality by design. In this term, in the protein aggregate analysis, three-dimensional homodyne light detection method, nano-tracking analysis, micro-Raman spectroscopy are going to be examined. For O-glycan analysis, we are challenging to development of new fluorescent labeling reagent. As intact molecule analysis that are useful for multi-domain products or conjugated products, we will have a comparative analysis of intact mass, CE, and ion exchange chromatography. For HCP, LC-MS-based method will be established. In addition to this evaluation for respective methods, we will try to how to apply the analytical QBD concept on these methodologies. I will mention this later. The latter half of the first term suffered from COVID-19, but before we had a lot of chance to communicate within the consortium. The activity of our consortium was published in the special topic commentary in Journal of Pharmaceutical Science in 2017. as the title of Recent Topics of Research in the Characterization and Quality Control of Biopharmaceuticals in Japan. In this meeting, Professor John Carpenter kindly joined and discussed the topics related to protein aggregates. We also reported in the JPS in 2020 with the title of Recent Achievement and Current Interest in Research on the Characterization and Quality Control of Biopharmaceuticals in Japan. At the meeting in 2018, Dr. Terekepari from NIHS and Dr. Raman from FDA joined and discussed. At the meeting in 2019, Professor Carpenter joined again. After this, we lost the opportunities to get together, but hopefully this year we can meet in person again. In Japan, we have Japanese pharmacopoeia as an official written standard. Different from US pharmacopoeia and European pharmacopoeia, JP does not have its own laboratories. So, our group contributes to prepare the draft document of JP general information and general tests based on the research in the consortium. It is good for us to prepare such documents with considering the implementation in pharmaceutical companies. In next few slides, I'd like to share the research topics on the protein aggregates. As you know, protein aggregates are prone to produce during the manufacturing process or storage periods of biopharmaceuticals. The aggregates have potential to induce the anti-drug antibody or immune cell activation. This may lead to the influence on PK, PD, or adverse events such as allergy. Therefore, protein aggregates need to be evaluated appropriately and strategy to reduce is necessary. However, there are issues on this risk management. One is analytical procedures for protein aggregates. The other is characterization of protein aggregates that induce immune responses. Protein aggregates potentially exist in biopharmaceuticals have size from nanometer to micrometer. Currently, the nanometer size aggregates are tested by size exclusion chromatography as purity tests for drug substance and drug products, and more than 10 micrometer aggregates are evaluated by light obscuration method in the specification of drug products as listed in pharmacopoeia. However, this evaluation is not sufficient, and other new methodologies such as flow imaging are desired. As you know, protein aggregates are transparent and have low refractive index compared to polystyrene standard particles that's used for the calculation of light obscuration method. Thus, light obscuration does not allow accurate quantification of protein particles. By flow imaging method, particles are defined based on contrast between particles and dispersion liquid. Thus, flow imaging has high sensitivity to detect nearly transparent particles. So, flow imaging method was thought to be suitable for evaluating protein aggregates in biopharmaceuticals. However, flow imaging method had not yet been standardized and not yet been listed in any compendium. Then, in our consortium, collaborative study to assess the adequacy and feasibility for the standardization of flow imaging technique was performed. We prepared 3 sub-visible particle preparations for the study. 2 was prepared from intravenous immunoglobulin by heat treatment, NPAS 90 and NPAS 70. Another one was ETFE particle kindly provided by Dr. Ripple from NIHS. The same samples was measured in 12 or 10 laboratories for LO and FI respectively. The purpose of the study was to elucidate the analytical performance of FI, mainly focusing on comparison among laboratories, comparison between manufacturers, MFI versus Procam, comparison between methods, LO versus FI. This is the image of the aggregate samples analyzed by flow imaging. NPAS 90 had low transparency, and NPAS 70 had high transparency. The transparency of ETFE was the middle of them. Preparation of NPAS 90 and 70 that were similar to protein aggregates and stable were critical to perform this collaborative study successfully. This was done by Dr. Shibata in our division. This is the result of evaluating the precision of particle count by using flow imaging method. 10 laboratories joined. Among them, 6 laboratories used MFI and 4 laboratories used FlowCam. Although there are some datasets with high variance. Overall, most of the median of particle counts in each laboratory were within 80 to 120% of the laboratory A to F or laboratory Z to J. This slide shows the result of measurement of these 3 types of particle numbers by LO and FI. As expected, the particle count measured by flow imaging was higher than light obscuration, especially for transparent samples such as NPAS 70 or ETFE. This means that flow imaging method is suitable for detecting transparent aggregates such as protein aggregates in biopharmaceuticals. In this study, we performed the comparison among laboratories, between manufacturers, and between methods, and revealed that the analytical performance of flow imaging. In conclusion, setting flow imaging technique as a release test would be feasible to precise analysis of the size and count of particles in biopharmaceuticals injections. It is conceivable that flow imaging technique is listed in a pharmacopoeia as a test method. Based on these results, new general information in JP was prepared and submitted to the JP Expert Committee in March 2022. In NIHS, we pursued the characterization of aggregates to identify the impact of biological activity. This slide shows the results of experiment to assess the relationship between size of antibody aggregates and Fc gamma receptor activation that are expressed on immune cells. By comparing the response of aggregates before and after filtration, we elucidated that the antibody aggregates less than 1 micrometer can activate immune cells via Fc gamma receptor. More detailed characterization is ongoing. We also identified the presence of silicon oil as a risk to increase the aggregate induced by agitation stress. As you can see, the presence of silicon oil increased the particle induced by agitation. As a result of enhanced formation of aggregates, the Fc gamma receptor activation was increased in the presence of silicon oil. Such kind of study may be useful to identify what are the risks for increasing the aggregate or identify the aggregates that have hazard for immune activation. And these observations can be used for risk mitigating strategies. As a third topic, I'd like to introduce the outline of the new project on next-generation therapeutic antibodies briefly. This project started last summer. Professor Tsumoto is a supervisor in AMED. The research topics are multi-attribute method using LCMS, Raman spectroscopy, cryo-EM, NMR, conjugated antibody evaluation and formulation screening, also bioprocessing of next-generation antibodies, and preparation of white paper and educational program. The first 4 topics are the main part of this project. Multi-attribute method using LCMS can analyze multiple chemical modifications simultaneously. Raman spectroscopy gives us conformational information in high concentration solution. Improvement of sensitivity is expected to enable further application. Cryo-electron microscopy will be used for evaluation of variation in 3D structure. Label-free NMR analysis using natural abundant isotope can be used for structural comparison. Integration of this analytical data should give us more deep understanding of product quality. Practically, this method will be used for process analytical technology or comparability assessment. As a last topic, I'd like to mention the perspective on analytical procedure development used for quality control of biopharmaceuticals in the era of analytical quality by design. Recently, 2 draft ICH quality guidelines were released and public consultation will come soon. One is a revision of Q2, validation of analytical procedures. The other is Q14, analytical procedure development. Multivariate analytical procedures that include spectroscopy are described in both guidelines. The concept of Q14 is also known as analytical quality by design. Quality by design can be said as science and risk-based approach for drug development. To date, quality by design was mainly applied to manufacturing process. However, from now, same approach can be applied to analytical procedures. Analytical QBD contains risk assessment for identification of parameter set points and range, then definition of analytical procedure control strategy. The aim of using analytical QBD is to reduce the risks to obtain inappropriate results through the life cycle and make it easier to the change management. As listed in the topics of the second term of Biopharmaceutical Consortium, implementation of the analytical quality by design is one of the key issues in next 10 years, I guess. This is a summary and future perspective. Risk-based approach in establishment of quality control strategy as well as analytical procedure development will be more important. Contribution of industries not only for product development but also for scientific aspects. For example, development of new analytical methodologies are desired, and these activities can accelerate the efficient product development. Regulatory science research can and should support them. Thank you for your attention. I sincerely appreciate my collaborators in NIHS and all members of Biopharmaceutical Consortium and Research Group on Next Generation Antibodies. Thank you.